Yeah, so I never took the plant propagation class at UC Davis. I always wanted to, and many of the faculty that really taught the very practical stuff have retired now. But anyways, I picked up this book when I was going to teach interns and for my lectures on plant propagation. I picked up this book. I highly recommend it. It's not particularly for succulents, but I always like to say that pretty much all plants work the same. Succulents just have some, because of their environmental, the way that they've evolved in the environment, some particulars for them, but all plants pretty much work the same. And so this book really helped me understand propagation much better and be able to explain it better. And since you're not taking the official class, you can buy the older editions, 6th and 7th editions. They're very relevant and much more affordable than the newest edition. I think they might be up to a 9th edition now. So in doing propagation, media oftentimes is important. Okay, it can be more important for some plants than others. I always joke that if it's a jade plant, do whatever you want and it'll probably work. But when we're trying to, for me, like I want to maximize growth and speed it up. And so I want to make roots happy and the tops of the plants happy, but media for roots is very important. And so on the upper left there is the mix that the most recent mix that we came up with at UC Davis in the last few years. And you can see lots of pumice in there. That's what we have available in California. A little bit of red lava. Okay, we were using red lava as a high amount in our mix for a while, and there were some serious problems with that. I think it had to do with it drying out too much and possibly some toxicity effects. A little decomposed granite. We used to have sand in our mix, but the decomposed granite is coarser. It's very small amount, so even if there's fines in it, there's very few fines in there, and then a little bit of organic matter for nutrient holding. The other compounds here don't hold much nutrients, and so if you grow in pure pumice, the downside is it doesn't have much what's called cation exchange, which is a reservoir of nutrients, which is important for plants growing long term and even roots as they're forming. So what we called our succulent mix, we'd cut it with the mix here on the left here. We'd cut it with one of these two versions of the pumice, the 3/16 or the 1/8 that we were getting from General Pumice Products in Southern California. Lately, I've just been at home. I've been buying Dry Stall, which is it's a more mix of pumice. It has fines in it as well, so it can be a little bit problematic in some cases. But that's why I like the sifted stuff. It's really good. So, and by the way, there at UC Davis, what we were doing is pasteurizing our media. We always pasteurized it before we grew in it. So sticking it in an oven of one sort or another and cooking it for at least 30 minutes to 180 degrees Fahrenheit. In small batches, you can microwave 5 to 10 minutes in a microwave. At home, I'm buy some turkey bags, and I'm gonna be sterilizing some mix for doing some propagation at home that way. So, because we're talking about propagation, vegetative propagation, these two hormones are really important. Auxin, which is produced up at the upper growing point of the plant. I have a whole lecture that I do to garden clubs of all sorts on hormones and how they work. But I'm just giving you the executive summary for propagation techniques today. But auxins are really important for root generation. As you can see up here, they promote root growth at low concentrations. And so especially our slow growing succulents, low concentrations is very important. Okay. But I'm going to show you in a little while how, produced at active growing shoot meristems and young leaves. So whenever you're taking cutting material, you want young expanding leaves and active meristems if you have them, ideally, and you will get better rooting. I'll show you some visuals of that. Okay. And then for the sake of branching, this hormone cytokinin produced at active growing root meristems. And so if your roots are happy, this hormone is produced more in the roots down there, and it travels up into the plant in the water transport tissue, and it promotes branching. If you know keiki in Hawaiian, it means baby, and branches, babies growing from the buds, etc. Cytokines will promote new branches being formed, and if you want a plant to branch, then promoting cytokinin by actually fertilizing your plant really well, you'll get more branching, and I'll sort of describe that a little bit more in a bit here. Okay. And for the panelists, if you guys have any questions as we're going along that come up that are relevant to discuss now as opposed to at the end, I'm happy to answer those, try to answer those right away. So please interject if necessary. So the auxin, these hormones, auxin and cytokinin, interplay with each other. Okay, and right here, this little arrow pointing up means high amount, arrow pointing down means low amount. And so right now what we're going to do is we're chopping the top of the plant off right there. We're taking the top away. So in this case, we've taken away the source of auxin because we've removed that. Okay, so we're pruning a plant right now. We took away the auxin, and now right up in here, cytokinin is going to build up. And when you have this combination of hormones, you get branching. Okay. And we all know this, it happens when we prune our plants. Well, if you're trying to propagate, get propagation material, and you want your plant to branch more so you can get cuttings from it, well, this is the sort of thing you could do. You could prune it to promote more lateral branching by taking away tips, and then you have more branching, and we're going to talk about in a second why these new tips are so important for propagation. Okay. And sometimes you get more than one branch growing because of the sensitivity of the plants to the combination of auxin and cytokinin. Okay, some plants branch quite a bit when you cut them, other plants just one new branch, and I'll discuss that a little bit more later. So when you have this combination of hormones, you get shoots forming, the upper part of the plant, leaves, stems, branches, etc. All right, well, there's a situation down here where you can cut a root off, and by the way, roots are going to form for the part that's still connected to the plant above that cut. Yes, the plant's going to branch because auxin promotes root formation. But right now I want to discuss this part, the root that was cut off, because we often think of that as just roots that are gone. But there's a number of plants out there that you can make root cuttings from. Okay, and what I know that you can do it from haworthias, some haworthias with the fleshier roots. Operculacaria is one that you can make root cuttings from. I haven't done a lot of propagation of Operculacaria, but anyways, root cuttings can be done. I don't know if they're better, easier to do than shoot cuttings, but there's a few plants out there. I'm curious to learn more about more things that that grow from root cuttings. So yeah, and by the way, this is what's done in tissue culture right here. This these hormone combinations to promote growing in tissue culture that I'll mention briefly because I'm not an expert in it by any means. But now let's look at the normal situation here. We cut a root off of a plant. Okay, we transplanted something, and you get root formation. Okay, well, if you want the plant to grow more on the top by promoting rooting, new roots forming, you're going to get growth. And so again, if you want propagation material, you can kind of wake up the plant sometimes by transplanting it during the growing season, and all those new roots will generate cytokinin, and you'll get a burst of growth in the top of the plant. So in this situation right here where we cut the root off, and above it you get a buildup of auxin, high amounts of auxin right there, low amounts of cytokinin, and that equals rooting. And where that really it matters to us is the situation where we cut right below a stem, and we get roots forming right there when we make cuttings of plants. Okay, and by the way, in that slide earlier that I showed you, the summary of auxin and cytokinin, auxin travels with gravity, and so if you take your cutting and you lay it sideways, it's not going to build up at the cut end over here. It's going to spread out across the plant like a cactus pad or a cactus stem that you leave laying down. And if you want better rooting, you want your cuttings nice and vertically upright, so that the auxin builds up at the very bottom end evenly, and you'll get much more evenly rooted plant. Okay. So, and again, I am not an expert on tissue culture. I know the science behind it, but one of the things that it takes to get really good at, especially tissue culture in particular, is lots of practice on the techniques, because what you're doing when you do tissue culture is you're taking little bits of cells of a plant, small little bits, and they're oftentimes susceptible to infection. And the technique is to prevent infection and pathogens from getting in there, fungal spores, bacterial spores. And so you have to get really good at what's called sterile technique, is working and clean. But, I've heard of people doing it with a fish tank on its side for Haworthia tissue culture propagation. So, a tank on its side, lots of alcohol to clean things off, and very sterile techniques. And that's what it takes for tissue culture. Okay. There's some tissue culture in the lab at UC Davis. I think those are strawberries or grapes, I forget. But we tried our hand at a tissue culture a number of years ago when I got a grant to do some propagation of aloes, and we could quickly move to just techniques that I'll show you a little bit later. But we tried our hand at tissue culture a little bit of some that were less available. We had less material for them, and there's infection right there of some kind of fungal pathogen that got in there. It's just practice is what it takes to get good at it. And when you have a grant, for only a year, and you're new to it, you may not be successful within that grant cycle. But someone that does do tissue culture, Tim Harvey down in Southern California, he has a tissue culture lab in his garage, nothing too complicated. But he has all the right tools to use sterile techniques, etc. And people are doing that a lot more with succulents. Beyond Tim Harvey at the Huntington, they have a big lab now as well. Okay, there's some of his stuff right there, including his little autoclave to sterilize things, and all the different hormones, etc., that he needs. Okay, so why can you do propagation of plants and even tissue culture? Okay, well, because you have these things called meristems, and I always joke that what's the happiest part of a plant? The meristem. Okay, and young cells that are undifferentiated, that are there to keep the plant growing, but insurance as well. So they're at the very top of the plant, the apex, but you also find meristems at the axillary buds, and as insurance. But anyways, these cells right here are all working together to form leaves and branches on a plant. So that's the top of the plant, magnified right here, and it's a cluster of cells, and they're all kind of, some are getting left behind and turning into leaves and other things, flowers at times, etc. But it's a group of cells. And if I can take those cells and I can separate individual cells or chunks of those cells and have them separate on their own, then the hormones, as I mentioned, the auxin and cytokinin, will interplay to generate a plant. And so you can either harvest this out and do a tissue culture, or one of my favorite techniques, I call it splitting the meristem. Some people go in and they drill out the meristems or cauterize it and burn it or something like that. But I like to just split it, and I'll show you how I did that. And we'll get back to that in a second. But one thing, going back just really quick, for the sake of auxin generation, these young expanding leaves up at the top of the plant are really critical, as well as the active growing meristem. That's where auxin is produced, at active growing, expanding leaves, active growing meristems and expanding leaves. And so you need that for good cuttings. And so right here, this is just to show you that I have some cuttings of the Trichocereus pachanoi, and then the Pilosocereus glaucochrois. And you can see the Pilosocereus, I used exclusively tip cuttings, and I found that they rooted much better. The pachanoi, these non-tip cuttings will root, but yes, the ones that are actively growing and tips they root much better. And I don't have good visuals of that, so I'll show you that with another non-succulent plant in a little bit. But again, I just want to point out that when you're harvesting cutting material, you want young expanding leaves and active meristems, because they're going to generate the hormones, the auxins, that are going to travel down and promote root growth. Yes, sometimes when we make cuttings, we take some hormones, different types of hormones, and we apply them to the cut end to initiate the process of root formation. But the continuous source from the meristem of the plant is actually going to have a greater effect. Okay, because when you add the hormone to the bottom, you're just kind of starting the process. You're saying, yes, you should become roots. But what really gets the root juices flowing? Not really, but what creates the roots to form and expand is the activity up here, the auxin, continuous source of auxin. Okay, and by the way, knowing and understanding these things over the last few years in the propagation they were doing there at UC Davis, we actually were using very little hormones, exogenous, applied hormones, synthetic hormones added. We were just using the plant, the source, natural source in the plant, because we were choosing these active growing, branches. We were taking cuttings when the plants were actively growing, when we saw that they were, young expanding leaves, etc. And we cut our use of hormones tremendously. Okay, and our succulents, especially the slower growing ones, you want to use, if you are gonna use hormones, you want to use really low amounts, like the 1%, which what Hormex 1 is. Rutone used to be able to get it with fungicide, which is great for like more delicate succulents. But there's other techniques to promote infections. But you low concentration. Don't use those higher amounts. And if you're like liquid stuff, you want to just put that on there and get it off of there like under the recommended time. Because if you leave a cutting in too long in hormone, you add too much hormone to the cut end, you can add lots of auxin inhibits cell elongation, and you'll just get nubby growth there, callus it's called, and you won't get root elongation. So you don't want to overdo it. It's better to underdo it. Okay. So it turns out that Hormex has what's called IBA, indole butyric acid, and a lot of hormones out there. That's what you get, the IBA, because it's the most active. But some of our more sensitive succulents, something like NAA or NAD, which is a less strong, less reactive hormone, auxin, is much better for succulents because it's milder. It doesn't overdo it, which is, as I mentioned, the problem of adding too much. So less reactive hormones like NAA or NAD are good for succulents. Okay, you can't get it with the fungicide anymore. Okay, so going back to the meristem splitting, and on PowerPoint you have to go in certain steps. So sometimes I reorganize my lectures sometimes. So anyways, in this case, Aloe-Palanzii, a very, what we call an apically dominant aloe that grows just vertically, several feet before it ever branches. And I want to show you how I took one by managing the hormones, if you will, and made it into a shrub, branchy. So I took here and I split it with a stainless steel knife. These are just some tongue depressors from our vet school that were that we recycled into plant use, and we've just used them as spacers after I cut. So you can see my cut right here in the center one, and then I just, after I cut, I put this spacer in there, because if you cut and then don't put it like something to keep the cells apart, the apical meristem, they will regrow back into each other and recover. And I have experience in that. And in this little situation here, well, this situation, I was playing around with cytokinin and adding it in at the axillary bud, into the axils of the leaves where the axillary buds should be, and trying to promote branching. And it turns out this aloe is so slow growing, so sensitive, that wherever we added cytokinin, it was bad. It caused problems. That's the summary right there. Okay, it was just a weird growth that the tissue turned kind of green, and nothing good happened really. So these were done, back in 2011, and you can see this green area here. That's where the parts where the cytokinin were added down the bases there, and it just caused the tissue to sort of just kind of swell up a little bit and turn green. It was kind of odd, but nothing good happened. Okay, so it wasn't a failure; it was a learning experience. Okay, Okay. So again, take great care to not overdo it with hormones, especially with your slower growing succulents. Okay. So here we are on the left is the one that if you see, you can read down there, it says no hormone. And this one was just split 4 ways and 4 branches grew. By the way, we try to be as even as possible, but because this is what we call a very apically dominant plant, one branch took over and became the mama branch that produced more auxin than these ones over here did and told them not to grow as much. And that's how the plant balances growth. By splitting it, we're just directing 4 branches instead of 1 branch, and a division of labor or whatever. So the one here on the right, the 2 that are missing there, because this was also split 4 ways, are the ones that I added cytokinin to, and they just kind of petered out. Branches kind of started, but then they died. They were messed up. They were just too much hormone addition. And by the way, and this is just this is ugly right here, right? No bueno. Okay, well, this is one of those like do as I say, not as I did, and I won't do again, which is in this case, I didn't use a sharp sterile tool. I used a dull, unsterile serrated knife, and by when I made this cut through here, I was showing some in a haste of trying to show some interns how to do this. I didn't practice good techniques, and I used a dirty serrated knife, and I introduced an infection into the cut there, and I lost that plant right there. So clean tools and sharp tools. Sharp is so important. The less cells you damage, I gave myself a little cut with a box cutter knife a couple days ago, about 4 days ago, and it's like almost gone, healed, because I used a very sharp implement for better or worse, and it caused very little damage, and the recovery was much quicker. So sharpen your tools for your cuttings, sterilize them, and you will have more success. That is the technique. Okay. A number of years ago, I back in 2017, I took the plant here on the left, and I split it again, and only the main one, and I got it to branch several times. We've since just before I left the university, a couple of these branches were removed, and they were rooting, and taking a long time, but the branches were removed to get some cuttings from this individual. So isn't that amazing? You could take a plant that's normally a single stem plant, and you can make it branch by splitting the meristems and having them go their own ways, so to speak, but still connected via the roots. Okay. So coring and etc. This is an alopalanzii on the left there that I did this to. I split it, and by the way, one time I tried to just use an awl, and I heated up high amounts, and I went in there and I kind of cauterized the meristem, shoved it in there, and just try to destroy the center of the meristem, but try to hopefully the axillary buds would recover. But because I went in there and just burned it and didn't probably swish it around and cause more damage, the plant actually recovered and grew back into itself. So I learned that you really have to separate the parts. And this is 2 of the 4 quarters of Alocaris bergensis there that I split. So and nowadays I just use clean new greenhouse labels that I put as spacers in between. So here's one, the Victoria reginae, aka the white rhino agave, very sought after out there. And I learned this technique from Rob Roy down in SoCal, how he did it. So I played around with it myself, and taking this plant and cutting it into pieces. These plants, when they're small, they oftentimes will here and there throw an offset off. And so you don't have to do much, but you'll get offsets here and there. But you want to generate more material, and this is also how Randy Baldwin down in at San Marcos Growers in Southern California does it, where they have a pretty good amount of these. Is you go through and you split the plant all the way through. Again, clean, sterile stainless steel knife, very sharp. I cut all the way through to the plant, and then I just took and buried the roots. So that to minimize shock to the plant, and it's already a slow growing plant, right? So buried the roots in there, but kept the above ground parts, the cut part above ground so it would dry and heal, which is very important. I'll talk about that more as I go. And then just waited, okay? And so, this is a little video of doing it here, but I'm just gonna speed up here and go to here so you guys can see this happen, okay? And there we are, taking the stainless steel knife. It's, agave, it's kind of fibrous, so it's a little hard to work through, so you can't just do a nice, smooth, clean cut, unless maybe I had like a samurai sword or something. Go in there, and there's the part, peel the part. And I was gonna do 4 quarters, but I decided to do a half and 2 quarters. Work through there and did that, and then let's speed up here. And there's the 3 pieces, okay? And, just trying to be as clean as possible. Agaves don't tend to rot very easily, so I could have been more clean, but I wasn't too worried about this because I found that agaves don't rot very easily. Typically, it's sometimes you might re-clean your tool between cuts, and which would be a safer thing to do. But anyways, there's the 3 pieces right there. I'll split it out. Sorry, I'll lay it out there. Okay. Okay, let me speed the next one here. Yeah. So go back for a second. So anyways, that's it did that there. And I think let's go back to this slide for a second. Oh yeah, so I thought I had the picture of it when it was when they rooted here, but I missed that in this lecture here. But anyways, it was about a year later after we did this where we started getting some branches growing and that we could then pull off or leave a little bit longer till we broke them off. But one of the pieces at least had multiple branches on it growing. But it was a year, and this is in a greenhouse, okay, under relatively ideal conditions. So pretty drawn out process. So Meristem splitting, a lot of people were doing it with agaves and aloes, and I thought, you know what, this should work on dicots too. Dicots, things like cacti, and split the meristem, because like on this Pilocereus glaucocrois, we used to use this a lot for grafting, and I'll talk about it in a bit. And whenever you cut something like this, only one of the little axillary meristems, one of the little buds in the areoles there will grow out, and you'll just get one branch. But see right here, I took this one and I split it, to get more branching, and sure enough, you can see there on the right how there's more than one branch from a cut point, because I took that meristem, took the top of the plant and I split it so that the cytokinin traveled to this one and traveled to this one, and then they each behaved independently, and the one that started growing first wouldn't tell the other axillary buds on these separate parts to not grow, and so it branched, and I got more cutting material out of it. And I've done this. I lectured to a variety of garden clubs, and plumeria people in Southern California were like, "Ooh, yay! Another way of getting more cuttings of our, clonal specific varieties of plumeria." So it does work. I will add that I found with the cacti, if you'll notice this one, I cut the top of it off. I found with some cacti that if you just split them and you don't actually remove the apical meristem so they regrow from an axillary bud, if you just split the top, sometimes they'll grow kind of sideways, not crested per se, but they're just asymmetrically for a while until they repair themselves, and so they'll grow kind of wonky, not in like where you get this very top of the plant that set cells apart. You just grow one-sided, and so I found this technique worked better for a number of cacti where you just cut the top off, and then you get branching from individual axillary buds in the areoles. Okay. So this is one of these situations where I was trying to speed up a growth of a plant, and this the Certanthus spiralis that I got from Ann Shine, an amazing grower and someone who was into copiapoas before copiapoas were like the new thing. Anyways, she had this Certanthus spiralis, and a really beautiful spiral leaf individual. Now there's a lot of plants out there that we know that have spiral leaves like this. But anyways, the bulb didn't branch very much because it's very apically dominant individual. So it didn't branch very readily, and I was watering it, trying to get it to speed up the growth. And our media then, it was just not very good, and I actually killed the main stem on this individual right here. And luckily, for better or worse, I didn't kill it all the way. And the little axillary buds that were at the bottom of the down in here, because there's the bulb is actually made up of leaves. There's meristems down in there. A bunch of them were released, and I got a bunch of branches growing. So it was a fortuitous accident, if you will. But I know that there's a bunch of bulbs where people do just like I showed you with the agave earlier, where they'll take the bulb and they'll split it and do something similar as I did before. I know somebody that he would take bulbs and he would split them, and he would put crazy glue in between them and glue them back together, and that way the wound is sealed up and healed, and he would just bury them, and then the plant would branch because there was effectively two halves. They were just glued together to prevent infection, which is a really cool technique. Now, I haven't done it very much, but there's some seedlings of Certanthus spiralis that we grew at Davis, and some of them were more branchy than others. And so, we cared about the curlicue leaves, so we wanted to vegetatively propagate that particular one. Okay, I mentioned, again, I want to show you visually the importance of active growing root meristems for better propagation techniques. And look at this, here on the right, this cutting that this intern had, young expanding leaves, a second branch over here, so multiple active growing meristems as well as young expanding leaves, versus the one on the left here on the screen that doesn't have any young expanding leaves and no active meristem. And you can see there's a meristem in here somewhere, but look at how little rooted this one, how few roots this one resulted in versus the one that was the good cutting material. And again, that's what I'm trying to emphasize here. If you want to, faster, better, using the better techniques, if you will, to do it. Okay, and we did this with Didaria madagascarensis as a more of a sort of deserty succulent plant as an example. Tip with active young expanding leaves, active meristem, versus these what I call subtips, the no apex on there, no active meristem. And most of these ones over here, they had leaves on them at one point, but they dropped other leaves, versus the tip here that kept going. And by the way, this is an exceptionally large cutting. In general, when I'm doing cuttings, my rule is 4 to 8 inches, so that you don't get something that's that falls over easily, or something that's too small and kind of susceptible to just not having enough in there inside of it to keep going. So, by the way, some succulents can be propagated on a regular mist bench with bottom heat, as we say, where it's about 75 to 80 Fahrenheit, and with mist. Things that have leaves on them. We actually did Dorstinia gigas under mist like this and had better results than having them dry. Why? Because those plants are facultative desert plants where they make leaves when it's wet and they lose the leaves when it's dry. Ocotillo the same way. They like moisture for growth, and so then they're very sensitive to dry conditions, and they'll drop all their leaves. And then if you drop all your leaves, less oxygen production, less rooting. So yeah, sometimes you can do cuttings under these are regular plants here, but we did and do succulents under these conditions like this here. This is an aeroponic propagation chamber. It's basically a tub with a little motor in it that's spraying water up into the cuttings that are floating in air, held upright by these styrofoam or neoprene collars. And this was developed for the 15 years ago for the hydroponic tomato growers, marijuana. but this technique works for a lot of plants, including Derstenia gigas right here. Okay, look at that. There's a cutting of Derstenia gigas. The thing is floating in air. That's what we call aeroponic, and then it's getting sprayed by the little micro sprinklers up into the cutting. And looking at those beautiful roots that were formed on that. And by the way, we had some problems with these working, and then we treated the plants with a fungicide and got much better rooting, because there was some, like, fungus that was kind of just taking away the vigor of the plants. So here's some that we did a couple of years ago. And what you see in the background, yes, those are tomato plants. And the tomato plants are in there. Back in here, the ones with the really big roots are tomato cuttings. And here's the Derstenia gigas cuttings over here, and some other plants in there as well. But the idea is that with these tomatoes, their roots floating in there and getting sprayed with the water, auxin was getting removed from those root tips and going into the water, sort of low concentration, and that was going to transfer over to the Derstenia gigas to get them to root. So I never did the control where I did with and without to really show if it really improved in situations. But hey, that's something that maybe I should do in a more controlled situation. Okay, so in the case of backing up for a second, in the case of making the Derstenia gigas cuttings, okay, these plants are plants, like I said, mentioned, that they're a desert plant. They are succulents, but they like water because they only grow when there's rain, rain and moisture around. And so for plants like that, I cut it, and I didn't let them heal. I didn't let them like dry, like the wounds heal. I just cut them and put them in this humid environment here to root. Okay, whereas you might do something like Eulychnia castanea here. I take my cuttings, I cut them off the mother plant, and then I put them upright in an airy environment, in this case in a flat with coarse, large holes in here, so the air can get underneath there. Sometimes I might even like have two flats, so that one stacked inside the other, so it's definitely elevated off the a bench underneath or whatever, so the more air can get in there. And you want this wound to heal really well and turn leathery. This is not ready to put into media. I always, for a great majority of my succulents, especially the slower growing stem succulents, I want to stand them upright and let the wound heal really well, so it turns leathery. Look at that, it's drying out, it's turning more leathery. And then if you look closely, you might start seeing some roots. There we go, there's some roots there starting to form. And now that's nice and leathery and white, so the tissue's all dried out and there's new roots growing. So now I can take this thing and put it into media. Okay, and again, the whole time, from as soon as I cut it to healing to ready to pot up, I kept this upright so that the auxin would travel down to the bottom and root. And for this one, I only did tip cuttings. Little side note that I learned from someone out there that grew castanea spiralis really well is that these plants with really good drainage, they can handle a little bit more moisture than some other plants, other cacti and succulents, and you can grow them a lot faster, but you have to have really good drainage. Okay. Anyways, leathery appearance to the wound for good root formation, to prevent infection, rot, etc. And so here's a bunch of them. I just laid them out for the photos, but you can see that just the wound is nice and leathery there. Okay. There's some other plants I would wait like months before I put them in pumice. Okay, so air dry, so that the wound turns nice and leathery, and only until it's turned nice and leathery and there's like really you don't see much green, it's just nice white there, then you put them in in this case, we were using red lava. This is during the red lava phase at the conservatory, which was terrible. It was terrible when we had high amounts of red lava that we're using. Is not good, and pumice is much better, but that's more of a story for another day. This is one that that I would let the cuttings sit for months, 6 months sometimes, of Euphorbia Abdul curry. Okay, so you can see how nice and leathery. When I did Abdul curry, when I took cuttings, my preference was to cut right at a pinch point right here. I know somebody, he said he would just take and cut the top off and do that, but we had enough material that we would only take the branches and wait till they turn really nice and leathery. And then I put them in pumice so that they continue rooting, because roots oftentimes will not form in sunlight or expand in sunlight. And so you want to put them in the dark and bury them in media, but only after they're leathery. So there they are right there. And this is the 1/8 inch pumice that we were using for these cuttings here. Okay. And just a couple more stem cuttings. This case, Tephrocarpus alexandriae, a really nice clone that we had at UC Davis. And if anybody out there has this, I'd love to do a little trade with you or whatever to get some of this back. If you have one that came from us. But anyways, I was just basically breaking off these branches. Sometimes I used a knife in here. It was hard to get in there, and so sometimes I would just snap it off, and just a quick snap so it would break and not pull the vascular tissue, the woody tissue out from the main one. So just snap them off. And then in this case, this situation, I was setting them just above pumice, of lava. It's actually red lava. I was just setting them above the red lava, just floating on the surface. And by the way, always upright. So I made sure that the one I'm pointing at the screen here, this one right here, that's the very top of the plant. All these are pretty close to the top and what was the bottom. Perhaps less of an issue because this is actually a whole branch, but I wanted to sort of continue on as it was. And with this one, what I did is I just took this, I call this the golf ball cactus myself. I took the stem cutting here and I just sat it in the amongst the other golf balls, if you will, of the Tephrocarpus and waited till I saw roots. And then when I saw roots, then I would hold it in a pot and pour pumice around it and plant it. So there it is right there, planted now. Okay. So these are some on the left, some degrafted Euphorbia pesidermis that were done a couple of years ago. I'm not sure what the result of these were, but the cuttings removed upright, floating in pumice. Okay. This is a really nice clone of Dorstenia levranii, another Dorstenia that likes moisture. And so I would take these cuttings, cut it off the plant, like I talked about with the Euphorbia abdul curry. But in this case, I would take these and just dab them on a paper towel until the sap that comes out of members of the fig family, the Dorstenias, bled out, and just keep dabbing it until it was done releasing all that sap. And then I would take it and put it into a succulent mix right here, or just straight pumice, and just keep watering them lightly but regularly, so to tell that stem underground that yes, you want to form roots because there's moisture around, and they rooted much faster than if I let them go really dry. So I'll go back for a second here. We'll talk about grafting as I finish up here. Succulents. A lot of things are succulents. There's a really large range of what we call succulents, and so some of them are very slow growing, like Euphorbia abdul curry, probably a facultative cam plant where it only does cam photosynthesis and very slow growing. You gotta be really careful with something like that. On the opposite end, you have jade plant. Do whatever you want. There's ones that have leaves on them. Anyways, you want to sort of shop around for the techniques that work well for different things, because they're what a succulent is just a plant that stores water, but there's a large range of habitats where they grow from, how susceptible they are to fungal infections, all that kind of stuff. There's general kind of categories that they fit in about how easy they are, how difficult they are, but techniques do matter. Okay. So anyways, here is, I want to show you the techniques of grafting. And really quick, why do we do it? To get the plant to branch or to survive outside of its own root system, because like Asticum hintonii and Asticum ritteri, super delicate with their roots, very sensitive, and can rot out really easily, difficult to grow on their roots, unless you have really good growing conditions, which is typically very hot for them. Anyways, grafted onto a Peresciopsis, actually a more tropical cactus. Here's Epithelantha micromeris, grafted onto Piloso cereus. And by the way, from the CSSA convention and you know, talking to people, I realized that Epithelantha is actually a temperate climate plant, and what are we doing growing it in our greenhouse? You know, when it comes from the southwestern U.S., might not be good in there. But anyways, Strombocactus. Look how branchy this one is because it was grafted onto this Peresciopsis, and it got lots of cytokinin, and that caused it to branch. And oftentimes when you graft onto something that has a more vigorous root system, generates more cytokinin, causes grafting or sometimes even kind of bloating of the plant because it gets, you know, loaded with cytokinin. So it kind of grows abnormally. But why do we do it? Because to keep the plants alive or to grow them faster, and then we degraft them sometimes, which I don't have a lot of experience degrafting, so please don't ask me about that. Anyways, I just wanted to, you know, there's a few different kinds that we did. For a while, people were using this, oh my gosh, Harisia juicebertii here on the left. We were using Pilosa cereus quite a bit. More recently, we started using Echinopsis. But you know, when you're making cuttings for grafting, you definitely want the tips, because when you graft, you only graft onto near the tip of a cactus, because that's where there's meristematic cells, so that you can get a better connection being formed. Okay. You can actually take and graft onto a non-rooted cutting. I've heard of people doing that successfully. Okay. But ideally, when it's already got some roots on it, is much better. But anyways, why do we do it? Well, you know, Astrophytum mysterious, 2 to 4 years to flower from seed. But look at this right here. This plant was germinated. This Astrophytum right here on top was germinated. You know, September of that 1 year. It was grafted 2 months later when it was like, you know, just about half a centimeter, 5 millimeters in size, onto this, in this case, Myrtillocactus. And by the way, people have stopped using Myrtillocactus very much because it's actually a slow-growing, a fast-growing cactus that dies really quickly. Any one branch on the plant dies pretty quickly, and probably from lack of water when you underwater it. And it's not a good long-term root stock. So much better ones out there to use that live longer. And the thing flowered within a year. Oh my God, this is like when Astrophytum were very unavailable, and now we have a flowering individual that we can pollinate and get more seed of, and grow them from seed and get more stuff. So, you know, part of, for me, propagation is to get more plants out there so that less people want to go dig them up from the wild. And so I'm all about bottoming out markets so that things aren't expensive. Anyways, Asticum hintonii, you know, right here, yeah, don't graft onto Myrtillocactus. You know, we learned that one, not good for the long term. So techniques of grafting, okay? It's actually, I've done workshops on this for a few succulent clubs, and most of the people, they tried, you know, they did 1 to 3 grafts, and everybody got one that worked, okay? And it's just being very clean about it. And it's actually not like tissue culture. You can be a little more dirty, but you still have to use sharp clean tools, okay? And so what we do is we cut the top of what we call the rootstock. We cut it off. And sometimes we shave the sides a little bit. And oftentimes you want to dome it so that you don't cut straight across, and then it shrinks, and then you get a little cavity where water can collect in there and infect the connection. So we cut the top of that one off, round it off. We took, in this case, the Epithelantha micromeris, and this is a crested plant. So the growing point, instead of being like this, it's more stretched out, flattened out, you know. I guess you could say like this, rather than a point. Okay, anyways, it's all stretched out. So all you got to do is you have to line up some of this ring in here. Oops, go back for a second. Sorry, clicked. Take this little ring here and line it up to that circle right there. Anywhere, just a little bit, and the connection we reform. Thanks to oxen. Thank you, oxen. And so they'll reconnect. So you just have to get them, push them up against each other, and say, "Hey, you guys want to be together in this relationship?" Okay. Anyway, sorry about that analogy there. And so you line them up, you put them on there, and then now you have to push them up against each other one way or the other. And I'll show you several techniques to do that, so that the cells connect up to each other. Okay, so rubber bands are the traditional material to use to bring them together. Okay, look at that, 2 rubber bands to make sure it was on there nice and snug. This is a more advanced technique. Rubber bands going in opposite directions. Okay, that's that, you know, the tension on that is you got to do it just right. Okay, but anyways, that was kind of my thumbs up right there. And so the next step is when you do the graft, you know, you've used, and I'm going to go back for a second because I want to point out something that I kind of forgot to mention, which is you can use the pruners to make the first cuts, a relatively not super sterile tool, but the final cuts, you use a nice, sharp, clean razor blade that it's either brand new or you took it and you sterilized it with alcohol, preferably, because if you use heat, you have to wait for it to cool down. But you cleaned it, and once it starts being anything kind of dull or you're not sure you can keep it sterile, get a new one, get a nice sharp, you know, for that final cut. So the final cuts are made with a nice sharp tool, and just like, you know, when you're cutting through butter, you don't saw through it, you just go right through there. Okay, to minimize moving infectious things around. Okay, so the clean, you know, sharp tools for your final cuts where you're going to join the two together, very important. And then the next step is to put it in a humidity chamber in some kind of humid conditions, just for a few days. Really, a week is really all you need, 7 days or so that those wounds don't dry up really fast, and the cells that in the between the two things don't dry, like when you put a bandage on, so you get less scarring. It turns out bandages, having a wound heal slowly on yourself, you'll get less scarring than if you just let it air dry. So you have it in a humidity chamber so that the two parts can connect up to each other, and then no more than 10 days later, you remove that cover off of there, pull it out of the bag, whatever, dehumidify so that the surface, the wounds don't start rotting on you. And that's enough. 1 week is the cells have made a connection. Okay. And there's the Echinopsis ones. Those were her practice. One of my interns, she was practicing there, and then when she got better, I had her do the Ostichium hintonii. And what I found with the Ostichium hintonii is they tend to callus at the underneath at the connection point with the rootstock, and so you oftentimes have to have them on there, like stretched on there tight longer, so they don't shove themselves off. This one was like, "You're not good enough for me," or something like that. So it happens sometimes. So other techniques, I was playing around with Teflon tape. Teflon tape is stretchy, the plumber's tape, the white stuff, and just stretch it over there, tape one side, and then stretch it over, you know, and pull it down, tape it on the other side, and boom, there you go. And you know, that worked out really well. There they are with the tape removed and outside of the humidity chamber. And you see how these wounds heal kind of slowly. You start getting a little bit of browning. If you get too much, you know, you've left them in the humidity chamber too much. But when they turn nice and leathery like this, like I showed you earlier with the cuttings, that's what you want. One of the students was playing around with, he liked parafilm. To me, the danger of this is that if you have any kind of, you know, fungal activity in here, you've trapped it in a very enclosed environment. But you know, doing things in this case, there is stretching the, you know, the parafilm over there to get it and hooking it onto the spines. Okay. And by the way, this was he was, in this case on the left here, he was doing Astrophytum caput medusae, the little long tubercles that are actually a stem. So there should be some meristem cells in there onto the echinopsis there. So I've actually saved in my career, I saved a few plants from rotting out. Something happened, probably because we're growing the epiphyllantha in the greenhouse, and it started rotting out. And so I went and found a little tip out here that did not have any of this red, any of this fungus that was in the stem tissue that hadn't made it out to the tips. I pulled a few tips off this giant plant and regrafted it and saved it. So sometimes that's the benefit of grafting. Okay, finish up with a couple more things as I wrap it up. So this is a little monster that I personally made a few years ago at the conservatory. This is an echinopsis at the bottom here, and I had grafted a Stechnium hintonii over here. This I forget this guy over here, this cactus. A few things on here at one point, and then I went back and added this Echinocereus reichenbachii, I forget the name of this guy, the Copiopoa, the small leaf one, I forget. Anyways, had a bunch of different cacti, and they were all grafted together. I had up to 10 cacti on that thing, and they lived for a while until the mother plant rotted out. We weren't looking. Okay, but look at this one right here. This is someone in Southern California did this or posted this, and they took, grafted it on this echinopsis onto, I think this is, I forget what this one is down below, and then a Pilosocereus, and then another cactus, and then topped off with a on the very top with a peyote or at least the genus Lophophora on there, and you can see what's going on here. And by the way, sometimes when these grafts are done, this one's grafted onto here, it'll be a little slower than the one it's grafted onto, and you'll get branches growing, and you don't want to leave those branches on there for very long because they will take over and they will keep this one from growing. In this case, this grower just, made a totem pole, a little totem pole cactus right here, okay? That's pretty cool, I think. So I always wanted to see how many different cacti I can get onto one, all together in one graft. And you can have quite a few, pretty much you can graft any two cacti to each other. Okay, as I finish up here, I'm gonna just do this and then finish up with some leaf propagation techniques. But I want to show you this little monster that someone made a number of years ago. And this is a graft where probably a Gymnoclasium milhanovicii right here, the moon cactus, if you will, was grafted onto. I think this is the Myrtillo cactus. And probably what happened is a cell from one, the Myrtillo cactus, and a cell from the Gymnoclasium where each had their nucleus, and those two cells were merged together as one cell with the two different sort of, the director's quarters of each cell, the nucleus, and then they're like in a relationship that they are not coming apart from, and they have to communicate with each other, and you get this, oddball chimera, it's called. Okay, leaf cuttings as I finish up here. There's a number of succulents that you can do leaf cuttings from. So you don't always need a stem with an axillary bud on there. Sometimes you can actually get, you can find meristems in leaf tissue. In this case, this is the base of the leaf here, but with gasterias, you can take strips of leaves and you will still get plants forming. You take that cutting. In this case, well, I'll show you right here with technique. This is Haworthia. We call them leaf pulls in this situation. You take a leaf and you take the plant and you pull the leaves off, and then we plant them in pumice upright again. And there's the plant right there before we did the pulls. And you take you don't do the very old leaves on the very outer edges if they're really old. You do the ones that are like, halfway through their life stage, if you will, and you pull those off. And the idea is you're pulling a little bit of stem tissue with them in this case, and then you're gonna root them. And there's a different species, Haworthia bayeri, but we did the same thing. We did the bayeri and the Comptoniana together at the same time. And there's my better pictures from the bayeri. with a little callus formed. That little callus, and it was a little bit of stem tissue, organized itself into a much better organized stem, and then you get branches growing from there. And eventually what we want to do is we want to separate these branches out from each other, but if you leave them all, because if you leave them all connected, one will take over and slow the other ones down. So this is Sansevieria kirkii. In this case, you can see this is one single leaf. There's the tip and then the sections down below. I actually dusted them with Hormex 3. I found that worked pretty well on these. Dusted the bottom, put them in pumice, and put them on our mist bench so that they got misted and kept regular moisture. I think I've had people do this bagged and it worked also. And then you get some plants forming right there. Okay. And look how beautiful those are, how beautifully rooted these new branches. And I will say this real quick. Don't ever try this with variegated Sansevieria because the new plants are formed from the vascular tissue, which is in the middle of the leaf, and the variegation is out on the edges of the leaf, and it does not translate to the cuttings, the leaf cuttings. So you will get all green cuttings. Okay. Anyways, this is Lachenalia, a South African bulb winter grower. We call it, our succulent growers grow these, and these are strips of leaves that are treated with hormone, put them upright in a mist bench, vermiculite perlite under that mist bench that I showed you earlier, and look at all the beautiful little bulblets that were formed at the base of those leaves. I think that's really awesome that you can do leaf cuttings. Okay, and speaking of awesome leaf cuttings, on the left of the slide here, not the Derstenia bullata, which is a normal stem cuttings, which is really awesome on its own, but these are leaf pulls of euphorbia, some of the Madagascan euphorbias, those low-growing euphorbias that are kind of branch kind of tightly. Turns out you can do leaf pulls. Like that's how Euphorbia françoisii is propagated, and a few of these other Madagascan. The student that I was working with at Davis was like, don't tell people about this because then everybody's going to start doing it. And I'm like, that's what I want to do is tell people how to do this so that more people can do this. Okay. This is another one that I learned about just recently in the last couple of years. As I finish up here, this is really my last slides here, is leaf cuttings of this Cyphostemma. And some of you guys probably may have seen this before or heard about this. This is not just Cyphostemma jute, this is Cyphostemma uter. Okay, these are leaf cuttings and leaf pulls, excuse me, not cuttings, because they were pulled off the plant, not cut. Leaf pulls that took a little stem with them. And in this case, this is the root stage where they're rooted. But what we did here, and the lines there show you about where we, we didn't cut, we pulled. We take those leaves and we pull them off. And in talking to people, I have not done this to fruition here. I left the university before I saw the results of this, but in talking to some people, if you do this sort of thing, these leaf pulls on like these, plants that are seasonal growers where they make their leaves seasonally, Cyphostemma here, you want to do this in just after the leaves have fully expanded, and in the early part of the season, and you pull them off, and then in this case we were using perlite on the left there, but we did it with pumice as well, and keep them moist, and you do it early in the season so that you get a nice stem forming before the leaf goes deciduous. It falls off like happens to Cyphostemmas during the winter months because they're summer growers. So you do these things when the leaves are just reached near full size early enough in the year so that you get a stem forming before again it goes dormant. I just want to emphasize that because if you do it late in the year, you might get roots, but then the leaf will fall off and you won't get a stem forming, which is really important. Okay, so sometimes you do like true leaf cuttings like Sansevieria gasteria, and other times you do pulls where you pull a little stem tissue along with the leaf, and then that itself is where the meristem is that generates the plant material. So yeah, I hope that this has been helpful to y'all, and look forward to the discussion here to talk about answer some questions for y'all. Thank you very much. But,
Thanks, Ernesto. That was great.
yeah.
Yeah, thank you very much.
You're welcome. I hope that it's kind of funny when you're doing this and like, you're not getting the interaction. So I hopefully people was mind juices were flowing back out there. But yeah, we can have a discussion here. I'll probably stop this and we can,
Sure.
yeah.
Thank you, Ernesto. That was wonderful.
My pleasure.
Yeah, I'm glad you finished up with Cyphostemma because that's a heavily poached plant. And knowing that you can do that with leaf cuttings with stem tissue attached, that's very important for people to know.
Yeah, definitely. Absolutely.
And for everyone in the audience, if you have any questions for Ernesto, please put them on the Q&A.
Okay, and I'm looking at the Q&A right here. So Bill Kinsey said, for those that can't get pumice, what can you tell us about vermiculite and perlite? Don't use vermiculite. Vermiculite, very bad for succulents because oftentimes it holds a lot of moisture, way too much moisture for succulents. Perlite is much better because it's kind of like pumice, but it holds even less moisture overall because it just holds it on the surface. Pumice is a little more porous. So pumice is acceptable. I mean, many years ago, actually, I took an Opuntia. It was Opuntia ficus indica, and I just put it in gravel from like the roof on my house, and rooted that. So you can just use some, coarse gravel. I would probably use, if I was using gravel, I did it one time with pea gravel. That's about 1/8 inch to 1/4 inch in size. So you have a little bit more surface area to trap moisture. Anything coarser is oftentimes too dry for most things. So it doesn't have to be pumice. It's something semi-porous that's coarse. Okay. Yeah.
Ernesto, I just want to mention the kind of illustrations you do in your presentation and the kind of difficult plants you're describing propagating. I think you have the making of a really good specialized propagating succulent book here, because that would be a fabulous reference for people, hobbyists that are getting into it. So they would know right away how to do something instead of trying trial and error on something that's already well known. And I think that's part of it's sort of a mystique with the succulent hobby, is that there's a lot of these secrets that people don't know. You could help a lot by making a book like that available if you would.
Thanks. I've thought about making maybe for starters some like YouTube videos to actually show the techniques a little bit more. Yeah. Thanks for that comment.
Sure.
Ernesto, how do you sterilize your tools? What do you use?
Alcohol, 70% alcohol, really good one. Little spray bottle, you can buy it, any, CVS or whatever, pharmacy, etc. And just spray this stuff down. I know people that will spray it down and then light it so that it burns off and a little heat to finish it off. Heat actually does work really well. Like, taking your pruners with one of those torches, like a little, creme brulee torch or whatever, or map torch, and you heat it up just for, a few seconds, burn things off. Those two work really well. If you use heat, you want to let it cool down before you start, getting into tissue. But those are the two major ways. Some people use TSP, trisodium phosphate, which is it denatures viruses and other cells. That works really well, but it oftentimes will degrade and eat your tools unless you have,
That's very corrosive.
Very corrosive. Yeah.
I'm just curious, what are some things that you've not been able to propagate yet with all your different methods? Is there anything that falls into that category?
Well, I don't know how those Euphorbia pesidermis turned out. Whether they worked or not, those are super slow growing. I know that they could sit there as a year and you might not get rooting. Let me circle back to that if I remember, so that
Okay.
I plant that seed in there for that. Yeah. Someone said, what lights are you using? They look red. Most of what I was showing you, we were doing it in a greenhouse where we had ambient light coming in from the outside, and then we just had LED lights for supplemental. So in that case, when you have some external light, just adding the intensity is all that matters, as long as you're not too far into the red. You don't want to use light that's very red when you're doing growing, because you tend to get plants that stretch out, because red light triggers stretching. You know, people argue about the quality of light for as you know, for better techniques for growing, but oftentimes intensity is if you have some amount of ambient light that adds a little bit of red light, etc., is the most important thing. So something kind of neutral is the best to use. I don't myself am not a fan of those like really orange, like purple or burgundy lights, or you know, magenta or whatever. It's not appealing to look at my plants that way. Yeah.
A couple questions about rooting hormones. Somebody wants to know about Clonex, and also how long these products last. When should they be discarded?
You know, that's a really good question, and I think that I know that, you know, we were using it for plants. We were using stuff that we stored in a cabinet under like warm conditions. Like it would get 90 in our workroom, 80 in our workroom, and you know, it was a powder for rooting, and it did work. It still worked. It wasn't stored under ideal conditions, so it keeps for a long time. They keep for a long time, unless probably if you have them out in the sun or something like that. They keep for quite a while. For succulents, I really like the powdered stuff better, because you're not adding moisture, which can, you know, promote, you know, if you trap some spores of, you know, fungi or bacteria in there, they can get into your wound. And so for succulents in general, the powder is much better, because you always put it on there and then you knock the extra off so you don't have too much on there. But so the powdered form, all those things, Clonex, it's all indyl butyric acid, unless you find the stuff that has the NAA or NAD. So look at your active ingredients. Super Thrive, the first version of Super Thrive, a fertilizer, had auxin in it, and I want to sort of play around with the one that has auxin in it to see if I can, you know, if that like the same nutrient concentration, but just adding a little bit of auxin, will that, you know, increase the root system and cause better growth in the plant. I see which vegetative propagation techniques, if any, are used by large-scale commercial growers. All of these. I mean, cuttings are one that's done a lot by commercial growers, like stem cuttings, because, you know, that's the material that you have. Some growers, the people that are really good commercial growers, they will actually have plants that are specifically for propagating from, and they will do things like cut them and then wait for new growth so that they have nice active growing branches, and then root those. So you're not getting like the one that's like semi-growing, the one that's like active growing really well. You want consistent, you know, consistency, and so you do things to have a lot of the same. Yeah.
I want to make a brief comment about tissue culture, because I did it for 2 years as a volunteer at Denver Botanic Gardens in a really good tissue culture lab. And despite all the precautions, we even used a heat device to heat up our tools, you know, to sterilize the tools by heat. We still got contamination from time to time. And one of the issues is it's very difficult, depending on what material you're working with, to sometimes get rid of the contamination that's inherent in the plant material itself. And so you always have to keep that in mind. Sometimes you do everything right and it still doesn't work.
Yeah.
And if you want to learn how to do tissue culture, I'd recommend volunteering at a botanic garden where they do it, because it doesn't cost you anything. They're always looking for people to do that. And you'll learn all the right techniques instead of trying to do it by trial and error yourself. I know people can do it in a fish tank, but really, you know, you're going to have some failures with that, especially if your technique's
Yeah.
not good.
And there are some videos online, I believe, of people showing you how to do it to get ideas. You know, YouTube is an amazing resource
Right.
out there for the world at large. So there are some, you know, information out there that way. Yeah. Let's see. Someone said tips for successfully growing Dendrocyckios. Well, Dendrocyckios, you don't vegetatively propagate it. You only grow it from seed, and that plant, it's very finicky. It would love coastal San Diego or coastal Central Coast of California. It's a very particular plant. You have to. It likes a narrow window of ideal conditions, if you will.
About Uncarina, somebody asked about that as well.
Uncarinas, I believe that those are done from stem cuttings. And so again, that's the sort of thing where you choose the cuttings earlier in the growing season, when the leaves are sort of like, you know, young and expanding, and probably keep them in a semi-humid environment rather than a very dry environment, because those are also plants that will drop their leaves when it's dry. And so this is one that, like, I convinced people like Woody Minnick when he was growing a lot of oh my gosh, the Ocotillo from the Fucaria purpusi. You know, I convinced him to water his Fucarias, and when he did that, he was like, "Oh my God, Ernesto, I got them to flower to seed much faster than if I'd have kept thinking that they were a desert plant that didn't use a lot of water." And so my point is, some things like Uncarina, a little bit of moisture is probably, you're going to get better rooting than if you keep them really dry.
Now, is there a better time of year for root cuttings?
Good question. Very good question. You know, I haven't done it enough to say yes. But you know, I know that with Haworthias, and I'm gonna go back to general here, is if you do it when the plants are actively growing at the beginning of their growing season, or during the height of the growing season, but not at the tail end of the growing season, across the board, you're gonna have much more success because wounds heal, there's activity in the plant, natural activity. You're not fighting against the environmental cues that tell the plant to shut down.
So you would say for just about all your propagation methods, go for the active part of the growing season.
Yeah. So are they winter growers? Are they summer growers? Are they plants that are triggered by moisture and that kind of keeps them going? You know, yeah, the active part of the growing season is the best. When we did that at Davis, our use of hormones, again, added hormones, way less, because we were just taking advantage of the natural cycle in the plants, if you will, to rebalance itself. Yeah.
David Schultz is asking about any experience with expanded shale.
You know, we didn't use that very much. I can't say. I would, if I was using expanded shale for my succulents, I would want it kind of broken up and chunky so that it would lock in a little bit more. Because when you have the nice round balls, they roll, right? And so if you have a top heavy succulent mixed into a mix, I believe it's sort of equivalent to pumice, if you will. But I can't say, I haven't played around with it very much at all.
I've used it a lot in my mixes and it works just fine. Yeah. But I don't think I'd try and use it straight for propagation. I wouldn't do that.
Yeah, those coarser particles, they're in general, they're adding filler. They don't have a lot of surface area. So they're really just decreasing the water holding capacity of your mix. And that's what they'll do. I mean, you could put some coarse rocks in your mix and lighten it that way, but then all of a sudden you can have a block that's too heavy.
What were the problems you had with the red lava?
Or scoria.
Yeah,
right?
the scoria. Yeah, scoria. Yeah, a geologist one time was like, "It's not red lava." The scoria. I think there was two things going on. I think definitely it's too dry, because scoria holds about 20%, 15 to 20% less moisture than pumice does, and oftentimes it's more coarse and it holds it more on the surface, at least the coarse particles. And so I think things just dried out too much. And what happens when you let your succulents dry out too much is that the roots die, and then the plant has to regrow root hairs, and you're fighting the cycle. And that's why you want to have a certain amount of micropores in your mix, because that's going to hold a reservoir of moisture that's not going to cause the plant to lose those root hairs that are really valuable for water and nutrient absorption. So when you use too much of it, that's the problem. It's too dry of a mix. Pumice has a little bit more of a reservoir of moisture, and it's not as dangerous that way as straight scoria was. Also, there's some stuff out there, very small amount of literature out there about it possibly having some toxicity effects. So I think it's mostly environmental conditions that are bad with just straight that, but it also, it might have been toxicity effects as well.
Some scoria could potentially contain trace amounts of heavy metals, and that might
Exactly.
be lead and zinc and things like that.
Yeah.
Here's a question I don't really understand, but maybe you will. It says, how do I maximize responses to BOCI requests?
Oh,
BGCI, excuse me, yeah.
I think I'm gonna say that that's probably a topic for another day.
Okay.
Botanic Gardens Conservation International. I think another question. I think we should move on.
Okay. Any other questions that the panel sees needs to be addressed? Let's see.
I see a question here relative to a woman has cans of Cormix powder from over 30 years. I wonder if they're still effective. You know, I used root tone forever and ever and ever, and I finally decided it probably the root tone wasn't effective in the first place. It was just the rooting I was using. It probably was no good long before I thought it was no
good. Right? Yeah. You know, I know that, like I said before, it's real quick. We used some old Hormex and we used the higher concentrations. We played around with different concentrations. They've been sitting in our cabinets for a long time, and they did work. Now we would always seal it up really quickly and put it away in the cabinet, so it wasn't out in sunlight. And it still worked. But like you're saying, Buck, technique, using the right timing, you're gonna need a lot less added hormone.
Hey, here's an interesting one. Have you added mycorrhizae to roots?
No, I haven't played around with that. You know, my thinking with succulents, and I'm not going to say I'm right, but my, you know, understanding is in deserts, you know, fungal activity is very low, and in general, fungal activity is very dangerous for succulents. And so I wouldn't go adding it very much, especially in a more humid, cooler environment. Maybe in desert environment where I can just trigger that activity just when, you know, when I add moisture, it might be okay. But I'd be really careful to start, you know, that's why we use lesser amounts of organic matter in our media, partly because it degrades and the pore sizes get smaller, but also when you have a lot of organic matter, you're encouraging an environment for fungal activity, and that can be dangerous to, especially your slower growing, more rare succulents. You know, jade plant, hey, do an experiment, try with and without, see if it actually works. But that's the thing that, you know, that's the thing is that we all have to do, and this happens a lot in horticulture. I'm just getting a little soapbox for at least 20 seconds here. We oftentimes do things and we say they work great, but really compare it to something else to see if this other thing worked better compared to this. And that's really good science right there. Otherwise, it's a little bit of voodoo work, like, and it might work, you know, but you really have to compare it to something else to see whether there really was a difference or not. And you have to do it in the right way to make sure you're really testing something, because we can convince ourselves in our lives that things work, right? So we have to be careful about that.
All right. Any final comments by the panel?
Thanks, Ernesto. Wonderful talk as always, and I learned so much from it.
Oh yeah, it was like it was a master class on propagation of succulents, is what it was.
Oh, well, thanks. It's you know, years of learning from other people as well. Okay, that's what is great about these things, is that we can share information with each other, and that's really what we should do. You know, what we do is in the hobby, right? We want to share information with each other, not be like, I'm the only one that does this. So, yeah.
Yep. All right. Well, thanks everyone. We'll see you all in 2 weeks.
Yeah.
Bye, everybody.
Thanks again.
Thanks,
Bye now.
Ernesto.
You're very welcome. Thank you.
Thank you, Ernest.
Bye-bye.